I'm an Associate Professor of Physiological Ecology here at SAMS, the Scottish Association for Marine Science up in the west coast of Scotland. So it is what I'm presenting here. It's work we've been carrying out over the past few years. So it's not just me in terms of the data, it's a big team effort over the years. So talking on behalf of the others as well. So this is Antarctic snow algae, and just to introduce the topic to this is a picture I took from the first trip in 2015 of Antarctic vegetation. So this is near the Rothera research station on Leone Island, and looking down towards the sea, you can see the green snow in the middle. This is green algae, and then in the mid-ground you have moss, lichen and grass, and then you have the red snow algae in the foreground. And this sort of largely captures, most of the major vegetation and photosynthetic groups that we have in Antarctica. So I'm really interested in sort of the function, the location, and the diversity and the physiology of these organisms. So if I grab some of that green snow and put it under a microscope, this is what it looks like. So you can see the microscopic algae are about 10 to 20 microns, and they're swimming around. They have large flagella, and so some of these are mating, some of these are dividing, and it's a very active ecosystem. So if I grab the red snow and put that under the microscope, you can see it's very sessile, and the cells are much larger. And if I just go in and out of focus here, you can see there's a halo around these cells as well. So this is like a sticky matrix that protects the cells for an EPS. So the cells are what many people consider as a dormant phase for this. So when we first started this work, quite a few number of years ago now, the current thinking about how this life cycle work would be the green algae would swim up through the snow in this slushy snow during the summer. And when they get to the top of the snowpack, there'd be environmental pressure, which would induce this hyperaccumulation of the red astaxanthin cell pigment. And then they would go into this dormant phase and then overwinter in this sort of encystment phase. And then this cycle would circle round and round and round. But we now know that this life cycle is probably the exception rather than the rule. So we wanted to work out, how much algae do we actually have on the terrestrial part of Antarctica from the photosynthetic point of view. We can't analyze all of it everywhere, so we have to rely on satellite imagery, so can we detect these blooms from space? Who lives there? What are they made of in terms of the physiology and the taxonomy? And are they the most abundant terrestrial photosynthetic organism in Antarctica compared to things like the mosses and the lichens? So our field locations, so we have the southern site on the Antarctic Peninsula down Rothera and Ryder Bay, and we have Robert Island, King George Island, the northern side there. So we work with we've been working with Inach and with Bass as well. So we have a mixture of data collection from the satellite imagery, from the field work and the lab work from 2015 to 2023. We also had the COVID years where we couldn't do anything at all. And from a satellite point of view, we heavily rely on the Sentinel work here. So you can see these image tracks using Sentinel Hub Playground. And if you look at the vegetation indices from, say, forested areas, you can see it's sort of yellow and blue and green. But what we're trying to do is detect these tiny bloom patches in Antarctica, which is hard, well, yeah, easier said than done, but we can now actually do this quite well. A study design, so these blooms here, so it's a typical sort of ecological study sampling technique. So we go into these patches and we do sampling in different locations, different patch areas, and we sample where there's visible snow algae and also sort of control sites where there's no visible sort of red and green patches as a negative control. There's a mixture of surface blooms as well. So some of these are quite hard and encrusted, especially if it's been sort of freeze-thaw cycles and it's very icy. But quite often you can't see it from the top, and we have to dig these pits going down there as well. So we sample different layers, like a cake layering effect from the top, going all the way down there. And there you can see the green layers and some of these red layers as well. Of course, we take a lot of metadata, the environmental metadata. So there's lots of the environmental parameters. There's techniques we can analyze actually on base, and also take measurements we have to do back in the lab as well. So we tried to collect as much data as possible in the field. And of course, one of the measurements we have to do in the field is the photosynthesis measurements to look at this carbon budget. So we use an ERGA, so it's an infrared gas analyzer. So basically, it's like a stethoscope we put on the ice cap or in the snowpack, and we know the CO2 concentration going in, then the CO2 concentration coming out. And of course, if it's negative, it means it's photosynthesizing, so it's taking up carbon. And if it's a positive difference, then of course it's giving out more CO2, so it's respiration. So we use these ERGAs to get a good measure of in situ photosynthesis and carbon uptake in the field. So we use this quite a lot. This is one of the ones on Robert Island, so it's quite difficult sometimes trying to find a grey box on a grey glacier on a grey day, but we do find them when we put them out as well. So we leave them out for about 12 hours at a time. So what we find is during the season, the blooms usually end up or start off green, and then they become sort of red during the season as well. And you can see this photo here on the bottom bottom right. You can see this nice cake layered effect as well. But again, this is probably more of an exception rather than the rule. And what we think is there's probably a range of species. So a species that are green all the time. There's species that are red all the time, and then there's species that can go from green to red and back to green again. So we have quite a mixed community in the field. And so this is from our 2020 paper, and you can see just a range of morphologies in this ecosystem. So we do put samples back, or take them back to the UK. So previously on the JCR, but also now on the SDA. And there we can do much more detailed analysis, looking at the metabarcoding of these snowpacks. We can look at the bacteria, the fungal, and the algal component to this. So it's quite nice getting a community composition. And then the other thing we start doing is isolating those cells, and so we get sort of pure cultures and strains, and we can start to do more detailed genetic and physiological analyses of those cells. So we start to sort of deconstruct that ecosystem, and we're looking at sort of differences in the physiological traits between those cells. And we also look at things like the metabolome, but I'm particularly interested in what's the metabolic traits for this. So we do GC mass spectrometry, for example, and we can start looking at the differences between how the function of the red cells versus the functionality of the green cells, and what type of metabolites you might need to sort of survive or exist as those two different types. So where we left off before this, the current sort of phase of the experimental work. So Andrew Gray, so we published this first map in 2020, and this is using Sentinel data from ESA, and you can see here trying to detect, using a mixture of the ground truthing work and the satellite imagery, where we're finding these green blooms along the Antarctic Peninsula. And because we have the field data as well, we can start to estimate how many tons of algae there is, the coverage in terms of kilometer square, and what that equates in terms of carbon, and how much carbon you need to fix that system. So we're starting to build up a nice picture of how much CO2 is sort of entering that ecosystem as well. If we want to analyze the red blooms are much harder to detect because the color looks quite similar to dust on the snowpack as well. But we can use, say, WellView 3 data and start to tease out these red signals as well. So we can start to see how the red and the green blooms transition over a season as well, and we get much bigger picture. So we know when we did the earlier studies that we are underestimating the coverage of these blooms. So this is very, very interesting. Of course, the what next? So this now is sort of kicking in more questions than answers. Now we've got a good handle of where they are. So the current project, obviously, it's just finished, was looking at the past, present, and the future of snow algae in Antarctica. So really trying to look at where these systems are and the functionality of it. So it's with us at SAMS, Edinburgh, Cambridge, and Bath, with a lot of input from INACH as well, from the logistics. So we have three main hypotheses looking at the controls of these algal blooms in the environment. Is it controlled by temperature or nutrients from marine sources? Do the snow algae actually fix more carbon within a growth season, sort of say per unit area, compared to the other vegetation? So how does it compare to, say, the moss and the lichen or the grass ecosystems? And with a warming Antarctic peninsula, how is that ecosystem likely to change? Obviously, with a warming area, these cells need slushy snow to bloom and grow. So if that disappears, or what effect might that have going forward with that ecosystem, because you're losing that carbon input? So for the fieldwork component, this is the bit I'll focus on. We chose Robert Island. So this is the top of the peninsula here. So a lot of you might be familiar with King George Island. So it's sort of two islands down from there. So you've got KGI, Nelson, and then Robert Island. And you can see it's largely a nice ice cap with an exposed land bit at this top corner. And you can see this picture here. So it covers a lot of the vegetation and areas to test some of our hypotheses and getting this environmental data. So it's got an abundance of vegetation. It's a nice marginal and a nice sentinel site here as well. And you can see a nice transition from to more established vegetation through to a lot of this sort of virgin territory where the ice cap is retreated. And then you've got the ice cap itself as well. And a lot of these remaining snowpacks where the snow algae can bloom, but also the ice cap where the ice algae can also exist. So in 2022-23, so we left Falkland Islands, went down to Rothera, and then collected equipment, analysed the algae there, then back up to Peninsula, got dropped off from the SDA at the end of December. So it's one of the first sort of terrestrial biology expeditions using the SDA. So it's fantastic to use that resource there and the logistics for the drop off and during the whole thing. And one of the key things was when we were going along the peninsula was the observation decks. So from the 2020 paper, we had a nice idea of where the blooms are, but going along the peninsula and then sort of ticking off to say, yes, we have a positive identification that there's blooms there, or there's evidence of sort of remnants of blooms along the way as well. So that's really, really useful. And also looking at, say, these penguin colonies here. So here you can almost imagine sort of a co-location of the penguins and the blooms here. So you can see the penguin pack at the top and the snow algae on the surface there as well. So you can imagine, the nutrient input from the birds going down to feed the algal blooms there. So if you haven't been, it's a fantastic place. We were incredibly lucky with the weather. So it was very, very calm most of the time. So anyway, but most of the time we're down in the ship. We prepare all the equipment for the expedition, all the packing. There's a phenomenal amount of equipment. So it drops us off on the on Robert Island end of December. And we said farewell to there as well. So there's 5 of us on the team. And sort of 2 to 4 staff from INACH as well. So it's about 9 of us living on the island over 2 months. So this is the base. This is the Riso Patron base. So it's run by INACH. So you can see the blue huts here. There's some historical huts there as well. The white hut is the lab. The accommodation is in the middle, and the food tents and the communication tents at the side there as well. So in the distance, we have an asper, and then the snowpack is looking the other way. So you can see here quite nicely this sort of nice great gradient in vegetation and snowpack going from left to right towards the ice pack. So this is what we're monitoring over those 2 months as the season progressed. So similar to the other sites, so we're collecting by hand, looking at these surface blooms, and here you can see the red tinge on the snow surface there. But of course, a lot of that is actually underneath the snowpack as well. So when you start to dig down, you can see this sort of nice green slushy snowpack as well. So again, it just shows the evidence that when we look at the satellite imagery, that we can't pick up a lot of these subsurface blooms as well. So we know we are sort of underestimating some of these the vegetation there as well. The other important thing we wanted to look at is where it's going. Okay, so we've got this snowpack on the top. So this is the terminal sort of ice cap area. So you can see the water sort of gushing out. Of course, that will contain a lot of the ice algae carbon going into the shore system and then going elsewhere as well. So one of the things we want to look at in the future is this sort of carbon load from the snowpack, from the ice algae pack, going into these sort of more shore systems. Okay. Yeah. So this is Charlotte with her drone. And Charlotte and Andrew had the drone, so they were doing a lot of work using these sort of Sentinel two sort of calibrated drone systems. So we had 2 to 3 drones. They were doing much more detailed mapping across the whole of the Robert Island site. So they were largely doing that. We had our satellite camps as well. So you can see in the ice cap here. So at the start of the season, the snow line was almost up to the tent. So you can see as the season progressed, the retreat of that sort of seasonal snow and ice pack moving further and further away. So we would sort of use this as a base during the day and then go out sampling from there. The other thing we're fortunate to have actually on the site there is this is the INACH lab. So we would have maybe a day of field work, then we have probably for every day we would have 2 days of processing those algal samples. So this is where we measure the snow chemistry, so the nitrates, phosphates, the ammonium, and then picking up those cells. We had we had a freeze dryer there as well, so we can actually freeze dry the samples so we can send them back to the UK for further analysis on, say, metabolomics, FTIR, and so on. So it's fantastic having this sort of resource on the INACH base to do all this. The other thing to flag up as well, just as an aside, is the amount of plastic pollution. I know it's been sort of in the media a lot the past year with Antarctic sort of plastic pollution. This is, as well as microplastics, we were finding a lot of macroplastics as well. So every day on this sort of shoreline, we were seeing a lot of these plastic waste being washed up on the shoreline there as well. So it's sort of plastic waste from looking at the bottles or from all around the world. So that's quite an interesting sort of thing that I've never seen before down there on previous trips. So on this site in particular, there's a huge amount of plastic waste being washed up. So we try to collect as much as possible for that. And I think INACH we're doing a study on the plastic waste as well after we collected it. So what did we actually collect? So we had about over 1,000 samples collected over the 2 months. So we had a lot of metabolite and eDNA samples, algal isolates, mosses and lichens. We have the gas exchange data, the hyperspec data for the imagery. So we had a really good understanding of the spatial and temporal study of these blooms here. So each dot there sort of represents a sample site from where we were. So come late February 2023, the SGA came back, the little Terror came along, and we spent the day sort of packing up and loading onto the ship. going back to KGI and then back to the UK. So what have we found out so far from this one? So one of the first papers that have come out of this is the surface darkening. So this is called the bioalbedo effect. So this is one of the ice caps here, and you can see there's sort of a grey, purpley, pink tinge to that surface. So this is largely ice algal cells. Okay, so as that darkens, the sort of hypothesis that you have a darker site, and that increases the heat load, so you get a slightly increased melting effect of that surface ice and snow. So if you look down the microscope, so we have these sort of microscopic probes we can actually put into the snowpack, and you can see here on the ice crystals exactly where the ice and snow algae are sort of stuck to the sides of these ice crystals in that sort of liquid water interface there. And again, if you look even deeper, we take the cells and do nice imagery in the lab. We're in the hut. You can see that, and you can see the purple traditional, I suppose, ice algal cells. This is the purple Antilonemia. These very bright sanguina and Clanomona cells, and Chloromonas, and these Raphidinemia ones. So this sort of diversity, we knew the sort of potentially Antilonemia was there, but we had the first time we've really sort of studied it and appreciated the abundance of the area that these purple ice algae actually cover on Robert Island, and in particular the sanguina as well. So this is again one of the sort of first studies looking at these big red sanguina cells at such an extent on this ice cap. So from the paper, so it's been published. So it's, I think it's about 2.7 kilometers square in terms of coverage of these ice algae across that ice cap. So it's about 6% of the area for the sort of photosynthetic life on the Antarctic continent. The energy budget, when we do the mathematics and the modelling from that, contributes to that 2.3%, Andrew and Alex calculated, of that ice melt, with that darker area contributing to that increased ice melt rate. In terms of the photosynthetic work and the net carbon exchange rates, so again, as well as the ice algae and the snow algae, putting the ergot on there, we also were doing ergoting on the moss, such as Sanionia, and the single grass species, the Dischampsia antarctica. So we had quite a good few number of days getting sort of this gas exchange, what we call net carbon exchange rates, from these different sort of ecotypes along the season here. So just a bit of a snapshot. So you've got grass, moss, and snow algae. So on the bottom three rows here, you have the light level. So there's a par meter attached to each one. And so you can see you've got time on the bottom here. So when it goes up, the sun is coming out. And if you look at the figure above it, so with the grass, for example, as the light goes up, the NCER value goes negative. And when it goes negative, that means that it's taking up more carbon than it's giving out. So it becomes more of a carbon sink rather than a carbon source. So we see, obviously, this is nothing new to science. If there's more light, there's more photosynthesis. But it's quite nice, measuring how this changes across those three types there. So we're measuring, obviously, the NCER over the course of those 2 months, and in these three different types here. So when we start to analyze the data, so say with the grass, if we put all the information together, say for Dischampsia, and we have the light level par on the x-axis and the net carbon exchange rate on the y-axis here. So it becomes a carbon sink roughly from about 500 par. So this is like 500 par is probably a day like today, it's a bit overcast. So it need about 500 micromoles of light for this Dischampsia to become a carbon sink rather than a carbon source. If you look at the moss, okay, a bit more of a complicated picture, but you need about 100, so you need much less light for that to become a carbon sink from what we're measuring in the field. Okay, so it becomes negative NCER after about 100 par. And similarly for the snow algae, obviously we have fewer cells per unit area, so the NCER rate is much lower. But again, if you sort of zoomed in, you would see you need about 100 par of light for when that system becomes a carbon sink rather than a carbon source where it's giving out more CO2, or in terms of respiration, more CO2 than it's taking up. So we can start to build up a nice picture of what sort of environmental parameters in terms of sunlight do you need for these ecosystems to become a carbon source or a carbon sink. And because we have information from Rothera in 2018, King George Island in 2019, we can start to compare these NCER values across different locations at different times, and looking at things like the environmental respiration and the gross environmental photosynthesis. So if you ever work with Charlotte, so Charlotte had a closed system as well on the island, closed erga system, so which is looking at much more detailed analysis of, say, the moss and the lichen respiration and carbon for photosynthesis values as well over time as well. But pulling the data together is quite nice seeing how the moss and the grass and the snow algae compares there as well. So you can see the Deschampsia is much more photosynthesis, but when you look at the total area, there's much more moss, much more Sanionia and snow algae compared to Deschampsia. So we need to look at this, how that compares on an area basis as well as a single species basis. We also start to measure things like total carbon and nitrogen, and we can start to again compare the nitrogen and carbon loads from, say, King George Island, Robert Island, and Rothera. And what is it? This is an interesting picture that came out. So Ryder Bay, this is near Rothera, these snowpacks at Rothera, and you can see the amount of nitrogen percent in these snow algal blooms we're detecting. But the delta 15N, this is the isotopic value, and if it's positive here, say it's almost just over 10, that implies that the nitrogen is coming from animal sources. So basically the animal feces, the nitrogen is coming from there and feeding the algal blooms that are happening at Ryder Bay. And we assumed that this sort of animal nutrient input would be the similar case for say, King George Island blooms and the Robert Island blooms as well. So where you have more animals, you would then have more blooms because it's feeding that nitrogen into those blooms. But that wasn't the case. So from KGI and the Robert Island sites, again, this is all analyzed on the same machine over the years. So we're very happy with the comparison over the years. Those sites, even though there were blooms there, the nitrogen isotopic values weren't indicative that they were taking up or necessarily needing that nitrogen from those animal sources. So that's quite an interesting observation that we'll delve into much more detail later as well. And again, and because we're at these sites over a long period of time, we can start to measure how the DOC load, so as well as the total carbon, we can look at the dissolved organic carbon and the total dissolved nitrogen load changes over that season as well. And apologies for the basic this sort of hot off the press data we were getting. So you can start to see the spread of the data over the season as well. And again, it's comparable to what we were finding at, say, Ryder Bay in 2018 from these snowpacks. So the DOC TDN, how it changes over the season in the snowpack where these blooms occur, is fairly consistent between the sites. Just checking the time. Okay for time. Yeah. The other thing we do I'm particularly interested in is the metabolic phenotype of these blooms as well. So from the biochemistry of these cells, how does it change, or what does it look like on a landscape level, and does that change over seasons and over years and over location? So one of the we have two techniques. One is what we call an FTIR spectra. So we get the cells and we drive them down and we put them on an FTIR scanner. And this gives us a snapshot of the type of other classes of metabolites that we might detect or we could see in those cells. So it's a non-destructive sort of fingerprint, metabolic fingerprint of the chemistry of those cells. So we can do this in the field, not in the field, or in the field huts. So we drive them down, put them on the scanner, and we get a snapshot of the metabolite. So as we do this, we compare this, we look at this picture over time, we can see for the biochemistry of those cells, we can start to compare, say, Ryder Bay 2018 to 2015, Robert Island in 23, KGI in 2019. So biochemically, they look similar. There's a lot of variation between the years, especially if you're looking at, say, something that's like 800 kilometers apart. So there's variation between years, and there's variation between sites as well. But that variation isn't necessarily greater between years or between sites as well. So it's the chemistry of these cells or this community seems to be fairly consistent. So that's data we can get in the field, and what's quite nice about that is if a sample, if we had catastrophic sort of sample destruction, from Antarctica to the UK, we actually have metabolic data that we can collect in the field. But the samples we bring back to the UK for analysis, we can do much more detailed sort of breakdown of the biochemistry of these blooms. So we have green blooms, mixed blooms, orange blooms, and we then extract the metabolites them through a GC mass spectrometer and start to look at which metabolites we're picking up in different algal bloom types. And we can start doing some fancy PCA analysis and looking at which metabolites are associated with being green and which metabolites are being associated with functioning as a red bloom as well. So we can start to build up a picture of a sort of which functional traits are important for each bloom type. So as we have this is the work we're currently looking at the moment. So this is what we call a VIP score plot. So the numbers on the right hand side here going from 2 to 7. So these are these are week numbers. So we're going from week 2 at Robert Island through to week 7. And so these plots here on MetaboAnalyst, this software, you can look at which metabolites are associated with sort of which time point they're being sampled with on Robert Island. So you can see here sucrose, these sort of carbohydrates, glycerol, these are sort of classic sort of cryoprotectant metabolites, are associated with building up in abundance in these communities over that time. And as well as the time, we can look at which metabolites are associated with each sampling point as well. So here we have a list at the top of different sampling locations on Robert Island, and again, which metabolites are associated with different sampling locations as well. So we get a nice sort of spatial temporal picture of those metabolites as well. Again, this is sort of work in progress, but it's giving us some nice clues. It's we get some quite usual suspects. Glycerol is the classic one that a lot of these metabolites organisms produce for cryoprotection. Yeah, so overall, so things we've been doing, in particular with Charlotte and Andrew and Claudia, building up these big vegetation maps. It's helping us build up these big vegetation maps of the whole of Antarctica, looking at snow algal maps, lichen maps areas, and anything that's green and in terms of vegetation across the whole of Antarctica. So this field work alongside the satellite imagery really helps us to build these models and also how they can sort of relate to these sort of conservation biogeographic region areas as well. So it's quite nice seeing how they compare in terms of the composition to these different locations. And again, the work that Andrew's been doing is looking at these sort of habitat suitability models and how that might change over the next, say, 100 years. So this is a paper currently in revision, but say got green algae at the top and red algae at the bottom. And I guess the two figures here is say the DNH figures, which is the change. So you've got the Antarctic Peninsula, and it's almost like a weather front sort of symbol there. These hexagons here. So where it's red, you see a decreased in terms of habitable snow for snow algae blooms, and if it's yellow, it implies that it's an increased habitable snow areas for snow algae. So it doesn't necessarily mean that the algae can actually reach there or get there, but we're looking at is a habitat suitable for those blooms to occur in the next 100 years. So it's quite interesting that we have this thing where the suitability is going further south, but also is going increasing in altitude as well when we look at the detail from there. So yeah, so going back to the original question then, how much algae do we actually have? So current estimates, it's about 3.3 kilometers square. So that's about 630 tons. Can we detect these blooms from space? Yes, we can do green and red, but it depends on the satellite and how much money you have. So the more the detailed resolution, the higher it costs. Who lives there? What are they made of? So we know there's genera like Chlorominema and Solanema, the Limnomonas, plus a huge array of bacteria and fungi. And we know there's variation in the temperature tolerances and these metabolic profiles. Are they the most abundant photosynthetic organism in Antarctica? No, it's unlikely. We have the current estimates. It's the lichens. There's about 8 kilometers square. The estimates from Charlotte's work for lichen coverage there at the moment. So yes, they're abundant, but not as abundant as the lichen coverage. So yeah, as always, it's a phenomenal team effort from everyone from logistics and Bass and Sam's and Match and everyone. So it's yeah. So thank you so much. I'll take some questions.
Thank you. That was fascinating. Oh, we have a hand straight up from Nadia. Would you like to go first?
Hello, Matt. Really nice talk. Thanks very much. In terms of the greening of Antarctica, you said that satellite methods have been able to better improve detections over the years. And there was a paper in 2024 about Antarctica becoming greener as a result of warming. Is that something that you have detected in the snow algae, or is there actually evidence for that, or is it just better detection in satellite?
Yeah, so going right back to where we started. So when we first started, we just had very observational, it's quite subjective data. These observations, they know that they're there, but there's been no systematic study. So we've gone from that to some evidence. We could detect them from some basic satellite imagery. So over the past 15 years, we've now got the better Sentinel data, the Copernicus data, and the well-through data. So the resolution has increased, and our confidence that what we're seeing is actually definitely algae rather than dust, which can look very similar, has improved. So, and we know that there's under snow blooms as well, so you can't see from space. So we know that there is an underestimate there as well. So in terms of is it changing by climate change, we don't have enough data yet to show from say the 70s when the first satellites were there to now from the snow algae point of view that they are changing. So this is one of the things we want to work on from that as well. I know there's other papers that have been out there recently to say it's changing from the other sort of vegetation point of view. But again, is that related to what's the ground truthing work as well? If you're solely relying just on the satellite imagery, you need that ground truth element there as well. Especially with things like if there's lots of cloud cover, as you know, from the top of the peninsula, there's a lot of cloud cover there as well. And a lot say that the moss goes green when it's damp and humid. So you might have a very green day, and a week later it's dry and you can't see it at all from the imagery. So yeah.
Yeah, it wasn't necessarily ground truth in the past. Do you think that they would be, if you had to guess which colour of snow algae would increase more under warming, would you have a colour that?
Good question. So from our lab studies, within the green, there's got 4 strains, and they all behave differently at different temperatures. So one hates anything above 8 degrees, and the other one doesn't particularly care what temperature it is. So I expect within the green, we'll see a shift in the community composition. The red cells they're a bit of an enigma, because they seem to come with, we think, from more sort of snow deposition as well. So they're probably seeded from one site and get blown onto another. So they're a bit more mobile. The green ones seem to be a bit more sessile. I don't know, but the green ones are associated with more coastal this sort of area. So we don't often see those at high altitude locations, but we do see the red ones at the higher altitude. So you could argue that maybe the red ones would survive better because the conditions will be more suitable higher up. Yeah, but we expect the low-lying flat ones near the sea will disappear, unfortunately. Yeah. And we see that elsewhere. In Scotland now, we've got very few remaining snowpacks in the summer.
Yeah, so true. Thank you.
Any other questions? Yes, go ahead, Yash.
Hello.
Oh, Yash, good to see you.
All right, nice to see you again. And great talk. I was wondering if you analyzed the eDNA data already using metagenomics or by metabarcoding, and if you found any other deeply diverging lineages in Antarctic habitat, because I guess it's not really well studied, so there might be some lineages, because I don't think many people have done it there.
Yeah, so we've sent the Robert Island data samples away for the metabarcoding and sequencing. So we're waiting for that to come back, and then we'll analyze that. Yeah, there's more and more groups. There's groups in Chile and the US also doing sort of metabarcoding analysis of these eDNA sampling of the snowpacks as well. So we are getting a nice picture of the fungi, the bacteria, and the snowpacks as well. But of course, the data analysis of that is only as good as, say, what's on NCBI or GeneBank in terms of the data.
Yeah.
So this is why picking out and isolating and characterizing the individuals, and then you can have more confidence when you do the eDNA analysis and things like on green genes and things. It is what that species is. So for the polar species, they are a bit underrepresented when you do the bioinformatics. So we need that more confidence in those taxonomic IDs of the individual strains. Yep.
Yep.
You can still get your beta diversity and the alpha diversity parameters, but it's but if you want to know exactly what it is, that's the isolation bit. And as you know, it's isolating cells is really, really hard. So.
Yeah. Yeah. Thank you.
Any other questions? I have one quickly, a quite general question. I couldn't believe how many different methods you were bringing together all in one go. It must be quite logistically challenging. I was wondering if you could just speak about what that's like.
Absolutely, it is. Yeah, it's obviously they say the more complex you have it, the more things can go wrong. It is careful planning. It's having a good team. It's having knowledge of the equipment. There's one season when I think the JCR was delayed, and so the equipment there's about a 4-week delay before the equipment arrived. So we had to sort of make use of what we had in terms of, rather than going back to basics in terms of having quadrats and just traditional ecological sampling. So I guess it's being prepared to do the sampling without the equipment is a key take-home message. We had a drone that broke, and things like that. So and there's power cuts and things. So but careful planning. It's having a backup plan, having a plan B and a plan C and a plan D, and trying to keep things simple. But at the same time, you trying to use the latest techniques. If you're doing DNA sampling, you've got to collect it in the right way, things like that. So it does add complexity if you want those sort of latest techniques to be involved in that analysis. But it's the logistics of getting everything there and back in one go is fantastic. This is where the INACH and the BAS logistics is fantastic for that.
Has high payoff for your research, I imagine.
It is, yeah. It's high risk, high reward. That's the thing.
For sure. Great. Did anyone else have anything they want to ask or in the chat? Oh yeah, Nadia, go ahead.
What's next? Are you going back?
Hopefully, yeah. So now we know. So this grant was, it's quite. So when we got the grant, it was in the middle of, it was 2021. So all the restrictions and stuff. So we were a bit delayed. So we couldn't do everything that we wanted. But now we know, got a much better handle of where they are, the taxonomy, and so on. So what we want to do now is, really homing in on the productivity of each species type. So there's productivity parameters of the snow algae, the moss and the lichens and so on, and how that productivity now might change over, the next 100 years in terms of the sort of vegetation load. So these are one of the things we want to test. The seeding, again, where these things are being seeded from, that's quite difficult science to do. And also these increased melt rates as well. So having these things like these autonomous Simba chains that can go into the snow, and as the snow melts and accumulate, we can measure that, and that data is sent to us back in the UK. So we get a much better idea of when the snow melts and these blooms are happening. So we don't have to be in the field all the time to do that, and we can put these out in more locations. So so I'm getting a much better handle of the productivity aspect. It's quite key for this, I think, because there's a lot of vegetation out there which a lot of people don't realise.
That's super cool. Well, good luck. Hope it goes well.
Thank you.
Thanks for organising, Lucy.
Cheers.
Great, thank you so much, Matt. And our next seminar will be on Central Arctic Ocean governance. So hopefully see lots of you there. But yeah, thank you all for coming, and thank you especially to Matt.
Okay, thanks for the invitation. It's been great. Thanks for the questions.